Hello friends, welcome to the new video. So we were discussing about the chapter heredity of class 10th biology. So today we will be continuing our topic. So here we will be discussing about these Mendel's laws in detail, their crosses, and this is our NCERT book as you can see. So we will be studying about these pages in detail. So let's start. As I have explained you about the first observations of the Mendel, that he discovered that if he crossed this tall and short plant, all plants were tall, and then when he selfed these all tall plants, he found that 3:1 ratio of tall and dwarf plants. That is, if 4 plants are there, 3 will be tall and 1 will be dwarf. So these were the Mendel's observations. So now let's continue our topic. Now here you can see any organism has two things. One is genotype and second is phenotype. Now what is this genotype? Genotype, as the name suggests, genetic distribution. What kind of genetic distribution is found in particular organism of a particular character, like tallness, color, etc. And here phenotype is physical character, that is which we can see easily, like a person is tall or short, he is white or black in color, like that. That is our phenotype. And genotype is genetic distribution, which we come to know through our genetic studies. So let's proceed. Now here I have written capital T and capital T twice. Now this is called as our gene, and these individual T and T are called as two alleles of one gene. Here both alleles are same, that is capital T and capital T. So this is the homozygous condition, that is both are the same alleles. So this is our homozygous condition. If one will be small and another will be capital, then this is our heterozygous condition, because both alleles are different. So this is the basic concept of alleles, that what are alleles and a gene. So now here capital T is dominant allele and small t is recessive allele. Now what is the meaning of this dominant and recessive? The one which will get expressed in homozygous as well as heterozygous condition. That is called as our dominant allele. That is, no matter if the allele small t is present with capital T, it will be expressed at any cost. That is our dominant allele. And if small t is present with small t, only then it will be expressed. That is dwarf plant. This is our recessive allele. That is, it will get expressed only in homozygous condition and not in heterozygous condition. Like when here capital T and small t, this plant will be tall because here dominant allele is present, so it will dominate over the small t, that is dwarf condition. So this is the whole concept about dominant and recessive alleles. So next we will be studying first cross of Mendel, that is monozygotic cross, or we can say monohybrid cross. Now firstly we will understand what is the meaning of this monohybrid. Here you can see I have written monohybrid cross, the cross in which only one pair of contrasting character is taken, like one character like tallness, shortness, that is height, one character, or like shape, round or wrinkled. And dihybrid cross is that in which two pairs of contrasting character is taken, like I will take height as well as color. So this will be the dihybrid cross. Now first cross of Mendel was monohybrid cross. So this is our monohybrid cross. This is capital T and small t, first cross of Mendel, and small t and small t. This is our long plant, and this is, or we can say tall plant, and this is our dwarf plant. So here we will separate out their gametes, like as we know that gametes are distributed as half, like small capital T and capital T are distributed, here small t and small t are distributed. Now there is a method to make this cross. These small t and small t will be written here, and capital T and capital T will be written here. So it is a easy method. This is our Punnett square which I was discussing in the previous video. So now here you can see this was, let's consider this is father, this is mother, or whatever you can consider as per your convenience. So here you can see that capital T and small t, these both combine to form capital T small t. Now this is their child or progeny. Now these are the four plants which were produced from these capital T plant and small t plant, that is tall and dwarf plant, their crossing. So this is our first filial generation. Now you can see that capital T is present in small t, as we know that small t is recessive allele, so it is not able to express itself in front of capital T. So here all plants will be tall. This is our phenotype, that all plants are tall, because tallness is a physical trait which we can easily see in a plant. But what is their genotype? Their genotype is capital T and small t. This is not easily visible as a tall plant. We don't know that it has this type of genotype or this kind of genotype, as these both are tall plants. So this is not visible by our eyes, and this is capital T and small t. This is the genotype. All these progeny plants, these are all are tall plants. This from this cross, Mendel proved this his law of dominance, that one allele is dominant and a second allele is recessive. Now what he did, selfing of F1, that is capital T small t with capital T small t. Now here we again broke this gametes like this, capital T small t, and here its gametes capital T and small t. Now as these gametes combine, these form the four kinds of plants. These are the alleles capital T, and two plants capital T small t, and fourth plant is small t and small t. Now capital T is one plant, this is two plants, this and this, and small t is one. This is our genotypic ratio, that is 1:2:1. Now what is phenotypic ratio? Pheno means physical trait, that is which is easily visible, that is tallness and dwarfness. So this is our 3:1. That is these three plants are tall, as we know that this is dominant allele, so they will dominant over recessive allele. So these three are tall and one is dwarf plant. This is our phenotypic ratio. So if you are asked about genotypic ratio, you have to write this for F2 generation, this is F2. And if you are asked about phenotypic ratio, then 3:1 will be phenotypic ratio. Now what are the observations? The characters which were not appeared in F1. Which characters didn't appear in F1? Dwarfness of a plant were visible in F2 generation. Which is visible in F2 generation? Dwarfness of a plant, that is small t and small t. So which shows that they do not show blending or mixing, and they are segregated. Segregated means separated in F2 generation. So this was an observation through this F2 cross of Mendel. Now this is called as law of segregation. That is, the factors which were not visible in F1 generation are visible in F2 generation. They didn't show any kind of mixing. That is, small t was not eliminated at all, but it wasn't able to express itself in F1 generation. And when selfing was done, it was able to express itself as a ratio of 1, and that was dwarf plant. So this is our law of segregation, where segregation means separation. So I hope you understood this second law of Mendel, that is law of segregation. We have completed two laws, first law of dominance and law of segregation. These both laws are derived from the monohybrid cross. Now in next video we will be discussing about our dihybrid cross, which will give us third law of Mendel from dihybrid cross. I hope you understood this very well. Thank you so much for watching.